Good morning, everyone. Welcome to the second day of the HCA Asia 2023 meeting. We had an academic feast yesterday, and we are poised to have another academic feast today. Still, our lectures have been lined up, and we start the day with a keynote lecture by Dr. Shin. The lecture will be chaired by Dr. Shyam Prabhakar. Shyam.
Morning, everyone. Welcome to day 2. And our keynote speaker this morning is going to kick us off to a flying start. Make sure this day lives up to the billing of yesterday as well. Shin, whom I think all of you know, on the organizing committee of the Human Cell Atlas, really a leader in single-cell technologies, one of the leaders of the Phantom Consortium, was at RIKEN for a while, drove a lot of single-cell technology there, and now I'm very thrilled to say he's my colleague at GIS the last couple of years. Going to give you an overview of single-cell technologies and applications. Thank you, Jay.
All right, good morning, everyone. I hope you guys had a good sleep, and I'm very excited to share with you some of the activities that been involving single-cell genomics for onset of my research career almost. So just going to give you a broad general overview of the field of single cells and where we are currently and where we are heading forward. And I think most of us here have seen this many, many times, and I think the ultimate goal is to understand the molecular makeup of these cell types in order that we can understand their relationship with one another. I think it's very critical. I like to give this analogy of an atom with protons and electrons, where a number of electrons dictates the identity of that element. And in a way, the number of genes and their expression patterns and their signatures also determine the functionality of that cell and gives that cell the identity. And really understanding this molecular makeup of these cells and how we can understand their gene regulation is one of the key fundamental questions that we need to understand, and that's one of the aims of my lab. And so how can we understand these regulatory features and these molecular aspects within the cells, and how are they controlled? And then once we understand their controlling mechanism, how can we manipulate some of these cells? And this really get into very interesting possibilities of leveraging single cells as ways for potential therapeutics in the future. But I really want to touch and emphasize the importance of gene regulation, which really is the fundamental process that influences everything in their cell fates, development, response to stimuli or infection of viruses, or even disease progression. And alterations in gene regulation can be really detrimental to the health and to the patients. So I love this image of gene regulation are multifaceted in that they can be regulated through chromatin level, at the chromatin level, through histones, or even protein complexes that bind to DNA methylation, to at the site of promoters or enhancers that can open the chromatin for transcription factors to bind and to activate gene transcription. So any dysregulation in these steps can really be detrimental, and further understanding how these regulatory processes work are extremely critical, not only for the identity of the cell type, but then their fates of the cell type. And I really want to start this by appreciating the efforts of Phantom that has made over the many years. And this was probably an effort, very first international genome effort that started almost 20 years ago, in early 2000. But when I joined RIKEN, the Phantom 4 was very much in full throttle, and they were really interested in not only studying the regulatory elements, but how these regulatory elements are interacting with one another. Because once you understand the one element regulating the other element, we can create edges between these two elements. And if you further expand that analogy, you can create a network or graph of all these multiple elements regulating one with the other. And that's precisely what Phantom 4 did. they tried to elucidate how monocytes can be differentiated into functional macrophages through understanding their changes in regulatory features and to build the gene regulatory network. And that's where I came in at RECAN, and this is already work done 10 years ago, so you can imagine how much of a single cell has evolved since then. But this is actually a single cell qPCR. Have you heard of that before? I know we are all very excited doing single cell RNA sequencing, but I have to tell you, at that time I didn't have a lot of money, so I couldn't do sequencing as a poor postdoc. And basically we wanted to investigate how these regulatory elements, mainly transcription factors, are altered in the process of monocyte to macrophage differentiation. And because of the effort done by Phantom4, we identified key regulatory factors, about 45 transcription factors, and we stimulated the cells with the differentiation regime and isolated 120 single cells at multiple time points. And again, this is an effort done many, many years ago, so I won't go into it in detail. But effectively, what we did is profile these 45 transcription factors at the single cell level in a dynamic differentiation process. This is the earlier days. This is even before the UMAP and TSNE was popularized. We were using multiple dimension scaling, and we were understanding how heterogeneous the cellular patterns were at the single cell level, only based on 45 transcription factors. And you could already appreciate these dynamic changes and heterogeneity of single cells during this differentiation process. If you were to pseudo-bulk them, you can appreciate the early response patterns to transition patterns and the completion patterns. And we were able to correlate a lot of these gene expressions at various time points and see this HOX-A genes with the CEBP was only correlated at specific time point, here in 24 hours compared to other time points, which meant that there were a time in this transition that their key genes are co-regulating. And so you can further extrapolate this across all genes that we profiled and identify a set of genes that are co-expressed, or we call them modules, and start to visualize how dynamically these modules are changing in the process of cellular differentiation. And you can see at 24-hour time point, a specific set of key genes are coactivities are increased, and at a different time point, they are lost, and completely new type of regulatory modules are reestablished. So what's really nice is that we are further able to understand what are synergistic and positive correlations, but which are also negative correlated. And you can bring these modules together, and we actually found one transcription factor, MIB, which is a proto-oncogene that are critical for monocyte proliferation, and that when we actually knock this MIB gene, we were able to see that a suspension monocyte cells were readily attaching to the bottom of the plate, suggesting that these are functionally changing to be a mature macrophages. And in fact, you do see a lot of the pro-differentiation markers activated upon the depletion of MIB. So I know this is a very old work, but I think a lot of these work still translates to this day to better understand how differentiation process are indeed regulated by multiple transcription factors and regulatory factors, and that there are key master regulators that are critical in the process of differentiation. And I think we are seeing more of these examples, of course, in iPS cells and other differentiation patterns. But we transition more into now let's go a lot wider scale and profile not only the monocyte differentiation, but how about across all tissues and all cell types of the human body? And that's really where the entry of Phantom5 was born. And the effort by Alistair Forrest and many other members also in this audience, like Piero and others, have really mapped out all the regulatory elements of the human genome by using a specific technology called 5' cage. And that allows us to do is to capture messenger RNAs that are capped at the 5' ends, and we can map them to the genome and identify a precise location of where the gene starts transcribing. We call this transcriptional start sites, TSS for short, and we're able to understand and infer where the promoters are and where the transcription factor binding sites are to activate this TSS. And what we also found was that these enhancer regions, a region where they are supporting the activation of promoters, were equally transcribing. And that using 5' cage, we were able to capture these enhancer-derived RNAs, or in short, eRNAs, and we were able to understand where the enhancers are in the genome, as well as where the promoters are. Now, once you understand these two sites or elements in the genome, we can further understand what are the transcription factors that are bound to these regions, and what are the key regulators in activating the promoters and enhancers, and even coordination of these two. So here's just an illustration of what I just talked about. You have a transcription factor machinery that binds to the promoter and activates the transcription at the TSS. Similar, same or different transcription factors can bind to an upstream or downstream of the promoter, which can then further activate and enhance the RNA. And both of these methods can be detected by 5' strategy. And we were able to we wanted to understand whether we can actually do this at the single cell resolution. So you remember the Fluidigm days, and bring you back all these memories here. But we used Fluidigm technology to capture single cells, and we developed a protocol to capture, again, the 5' ends of the RNA, and to understand where we can actually detect these transcriptional start sites, not only from the promoters, but also from enhancers. And here, what you're seeing in this bimodal plots are the signature of what enhancer RNAs look like. They're actually bidirectional, one going sense direction, and the other going the anti-sense direction. And you can beautifully see this pattern in bulk cage technology, as well as C1 Fluidigm-based single-cell RNA-seq method. But what was also interesting to us was that these enhancer RNAs are actually not bimodal. They are bimodal when you average them, but if you look at the single-cell resolution, you can see that there's one strand coming from sense direction, another strand going from anti-sense direction. So suggesting that you could only have one active transcription of one sense and not the other at any given time, and that only in a bulk you see the bidirectionality. But we were also able to visualize a lot of these enhanced RNAs using single molecule FISH. And this will be another potential area of interest for spatial biology, of course, is that we can actually map these enhanced RNAs and visualize where they are transcribed. And obviously, these are very nascent and very not stable transcripts. And so they're readily degraded. However, these nascent transcripts can be captured at single molecule resolution and co-localized with their nearby promoters, indicating that these are indeed co-activated with the promoters nearby. But we wanted to further expand the scalability of this approach by adopting 10x Genomics and employ their 5' chemistry to capture the 5' ends of the transcripts at a much higher scale. And to do that, we tested both poly-DT approach with capturing the long poly A tails, as well as random priming. And we wanted to ask whether we can visualize the difference of the gene expression. So here at the top are the 3' RNA-seq of MYC expression. And you can appreciate the very strong 3' ends activity. And most of the 3' single-cell RNA-seq that you perform, you're actually measuring the counts of these ends. When you apply the 5' chemistry, you can appreciate the sharp transcriptional start site peaks that are highly co-localized with the referenced MYC gene. But you could also appreciate another alternative transcriptional start sites of MYC just downstream here, which could potentially lead to various isoforms of the 5' ends. But what we appreciate is that there's actually not much difference between the poly-DT and random priming, of course, in the messenger RNAs. The question was, do we see any difference in enhancer RNA detection? So here, when you compare the 3' chemistry with the 5' chemistry, this is a pretty good correlation in terms of their gene expression patterns, despite that they're mapped in a very different location of the gene. And when you compare the 5' poly-DT strategy with the random strategy, we also see pretty strong correlation, surprisingly. And I think one of the reasons for this is because a lot of the poly-DT priming actually not only happen at the 3' ends, but there are a lot of internal priming process that takes in place because there are a lot of stretches of A's in a gene body, and they tend to be hybridized to poly-DT primers. So that allows us to capture actually many poly-A minus transcripts, such as enhancer RNAs. So here's just an example of using 10x 5' strategy. You can see NR4A2 locus and transcription of that gene going antisense direction. This is the promoter transcribed CRE, and they co-localize very well to the attack-seek or the open chromatin regions. But just upstream of this, which is not annotated by GenCode, have a strong bidirectionality patterns, which are, again, indication of enhancer RNAs. And they co-localize with the open chromatin as well. So we are, again, able to use poly-DT 5' chemistry and still capture these very nascent enhancer RNAs that are being transcribed just upstream of this TSS, which is, in a way, a multimodal strategy where we don't have to use attack-seek and 3', but just using one 5' chemistry, we're able to annotate both the promoters and enhancers of the same single cell. So here's just an example of expression of the promoter and the expression of enhancer RNAs of this promoter, which co-localize very well, in this case, PBMCs. But in order, because many of these enhancer RNAs are not found in GenCode, they need to be annotated. They need to be functionally annotated as a regulatory element. So Chung and I, we developed this software called SCafe, and you're able to run this using the 5' single cell chemistry, and it'll give you an annotation of their transcribed CREs. And this will be used for a gene expression quantification of both promoters and enhancers. So here is in the proximal, which are the promoters, but you also identify and annotate these distal transcribed CREs. These are enhancer RNAs, and they again co-localize very well with the attack-seek peaks in green. So now that we had this really nice technology under our belt, the question was now, can we expand and scale this strategy across the human tissue? And RIKEN is not a medical institution, so we had to pack our bags and travel across Japan and give this roadshow and say, hey, look, guys, we have this technology. Please give us your samples. Would you like to collaborate? And to our surprise, many of the clinicians agreed. When I first started this effort, many of the traditional scientists were saying, Jay, don't even bother. Clinicians not going to collaborate with you. But I didn't care. I just went around. I just wanted to travel across Japan and visit all these beautiful sites. And after the seminar, I was blown away by the amount of interest and willingness for clinicians to engage with our efforts to profile single cells. And the reason why it worked was that many clinicians really care for their patients, and they really want to understand their molecular basis of the samples that they're being derived from their patients. But what we were interested in from a genomics perspective was how we can take various tissues and integrate them to make the map. So I would have to say this was a really win-win collaboration where each clinicians were benefiting from this data, but at the same time the genomics lab really benefited from the integrativeness. So this is a summary of all the efforts that we've done. Chung had very nicely presented this yesterday, so I won't go into it in much detail, but we were able to collect and profile 23 tissues and about 400,000 cells and identified about 200,000 these transcribed CREs. And the question was, can we then use this map to then understand how regulatory factors, such as transcription factors, can mediate the binding to then manifest into a particular disease. But what's very important to understand is that the genetic variants are highly contextualized because if they are in the presence of right cell type, will give a disease. But even with the same mutation, but if it's not activated in astrocytes, it will not give rise to that disease. So in other words, if you have a transcription factor binding to both promoters and enhancers, you would have a normal homeostatic network, which gives you a very nice flow of outcome. But if you had mutations, either in enhancers or promoters, the transcription factor will be inhibited, and this will disrupt the network and leading to unwanted consequences. So we wanted to study that by mapping the TCRE atlas that we've generated together with all the GWAS summary statistics that have been done over the past decade or so, and co-localize the activities of TCREs with all the SNPs to ask which SNPs are relevant in which cell type. And here is a summary that there are various disease traits that are highly relevant in immune cells, and a lot of disease or normal behavioral traits of the brain are highly enriched in this. Neuronal cells and many of the stromal cells are associated with these various traits, including blood pressure and so on. And now this is the power of single cell is that we can go further into higher resolution, that you can subdivide and ask what are the subsets of T cells that are relevant for various disease traits. And so here's just a summary view that you can actually visualize at the single cell resolution which of the cell types or subtypes are relevant for various traits, such as psoriasis, are highly enriched in T cells, dendritic cells, and endothelial cells, where arthritis are different types of T cells, B cells, and melanocytes. And we can really further systematically understand the relevant and identify the relevant cell types that are critical for the manifestation of the disease phenotype. So this is a very busy slide, but I just want to highlight that we did identify many, but one of the examples are this enhancer element, DLO248, which is upstream of this promoter. And what's interesting is that there is a SNPs that are changed from A to G, which are very important for a transcription factor binding site, or RAF1. And so alteration in these SNPs can lead to a dysfunctionality of this transcription factor binding to that enhancer, which then alter the expression of promoters. So I hope that the first part of my presentation that talked about transcription factors and how they can create regulatory modules, but the real genuine activity of these transcription factors do bind to promoters or enhancers to elicit a downstream activity, but SNPs or any genetic variants can also lead to alteration in the transcription factor binding activity, which can potentially lead to disease phenotype. So, before we are able to associate only SNPs to disease traits, but with the power of single-cell genomics and systematic profiling of the human cell atlas, that you can actually identify the regulatory elements and define the gene programs and what are the cell types that can manifest into disease traits. I want to continue on the theme of gene regulatory network and how we can actually understand this process for potential regenerative medicine and stem cell research. And this was a side project that I was very much interested in when I was in RECAN. And this really comes with the notion of Conrad Waddington, who proposed that differentiation is a process like a marble rolling down into a hill with valleys and mountains. And what you can see is that depending on these junctions or bifurcation junctions, that there are key transcription factors that dictate the direction and the fate of where these cells can go. But now we know that we can completely disrupt this by overexpressing different transcription factors, such as Yamanaka factors, or direct the transdifferentiation from fibroblasts to a different cell type. And there you go. And one of the key question is, what is the regulatory network that control the cell fates? And can we understand these cell fates to transplant this into another cell type? So here you have, we also came up with several methods called Mogrify to understand what are the transcription factors that could be relevant in differentiating not only from stem cells to specialized cells, but from one specialized cells to another specialized cells, or transdifferentiation. So again, this is a work done many, many years ago, over now 11 years ago, so I won't go into much detail, but I want to bring you back to the original gene regulatory network of monocytes, and we asked whether we can transplant this monocyte regulatory network onto a dermal fibroblast, and can we reprogram dermal fibroblast to function like a monocyte or macrophages. So to do that, we got a pool of transcription factors that are defined by monocytes, and we overexpress them into a fibroblast. But because this is a sheer stochastic random event, you'll get different combinations of transcription factors that gets expressed at the single cell resolution. And now we can leverage the single cell technology to identify which of the elements here are co-expressed in these single cells to elicit the function of monocytes. Again, I didn't have money, so I used PCR, single cell PCR results to understand we were able to sort CD14 and HLA-DR expression in fibroblasts, and then identify which of the modulators are highly enriched compared to cells that are negative for CD14 and HLA-DR. Again, CD14 is not a fibroblast marker, it is a monocyte marker, and that we are able to activate these gene expression by single-cell RNA sequencing. And when we overexpress these two transcription factors, that we're able to elicit a phagocytic function in fibroblasts. Again, these phagocytic activities are observed in macrophages, and that we are able to elicit a phagocytic activities in fibroblasts by reprogramming the activity by overexpression of key modulators in the cell. But we want to scale this to another level and ask, can we do this across different cell types, mainly in the brain and the neurons, and identify which of the regulators of transcription factors are important for various subtypes of neuronal activities. So to do that, we performed a single-cell RNA sequencing of human brain organoids, and we performed a dynamic time course, and we were able to identify key regulatory factors, performing motif analysis. And we built a gene regulatory network of a developing brain. And then we asked, can we transplant this regulatory network onto a fibroblast and identify which of the regulatory factors are important to induce cell reprogramming at the single cell resolution. And we were able to then reconstruct this gene regulatory network for dopaminergic neurons or glutaminergic neurons or serotonergic neurons. So this, again, is a real power of using single-cell genomics, is that you're able to perform this experiment only once and perform all possible, or not all possible, but enriched combinations could be derived from single-cell RNA-seq. So here is just what I illustrated. You transplant this regulatory network by pulling these transcription factors, and then we perform single-cell RNA sequencing. But before that, you can already appreciate the vast changes in cellular morphology because you have now different combinations of transcription factors at the single-cell level, and different combinations give rise to different morphology. And we can do single-cell RNA sequencing and then identify the key transcription factors which activates the modularity of that specific neurons. So here's just an example of different cell types that we were able to reprogram. These are non-reprogrammed cells, which are highly enriched in fibroblastic genotype, fibroblastic molecular signatures. But on the left-hand side are highly activated neuronal, and we can further subdivide them based on their character. And they do follow a similar trajectory patterns as you would expect in a developing brain in the human fetus. You can also project in the pseudotime and understand what are the genes that are switched from fibroblastic to neuronal specification, and then also infer what are the transcription factors or gene ontologies that are modulated during this transition. So now that we could identify different subsets of neurons, we can ask what are the transcription factors that are enriched, and then indeed we're able to find PAX6 or NeuroG2 activating many of the neurons involved in this cholinergic neurons. And then systematically, we can then identify a set of transcription factors that are associated with glutamic or GABAergic, cholinergic, or dopaminergic neurons. And what's interesting is that this is nothing new. Many of these transcription factors have been previously reported, saying that these are key transcription factors in the differentiation of glutaminergic neurons, or DLX1 and 2 are critical for GABAergic neurons. But our platform of performing single-cell screening is allowing us to recapitulate and identify the key drivers that determines the cell fate in a very systematic and very simple approach. And interestingly, PAX6 is one of the key transcription factors important in neuronal differentiation, especially in neuroprogenitor cells, which were very critical in order for this reprogramming to take place. So this is my last slide. I just want to summarize that I've hopefully illustrated this efforts to profile many tissues, but I think there are a lot that needs to be done in order to reconstruct these gene regulatory networks. And I think we looked at cross tissues. There have been efforts to do across population variations. So there have been many EQTL talks. I think disease are absolutely critical. I think we're moving more into treatment responses and perhaps gene therapy or gene manipulations or regenerative medicine, for that matter, in really leveraging single-cell technology. And I think for translation is that AI and precision medicine will be further enhanced, and that hopefully we can come up with even more accurate strategies for tissue targeting. But I really want to summarize by highlighting this figure, is that I've talked to you about single-cell qPCR, and talked about various approaches of understanding gene regulatory network. I think this effort is going to continue and grow at much scale. And reason for that is the cost of genome sequencing is dramatically going down. As we all know, it's faster than the Moore's law, and that in expectation of additional sequencing companies that are coming out at the moment, I think this cost will continue to drive lower. In parallel, the technologies and capacity to profile single cell is only going up. And we talked about from PCR level to 10x genomics, and split-seq strategies, which allows 1 million single cells from single experiment. And spatial technology is actually allowing us to profile even more cells in a single experiment. So this divergence of increase in single cell profiling capabilities with decrease in sequencing cost, we can only expect that this number it will only increase. So really the problem is no longer whether you can profile single cells, but can you actually compute, and be able to process large data sets. And I think that's an area that's going to be very, very fascinating going in the future. But I also want to highlight really the benefit of why single cell is going to be really powerful for elucidating not only gene regulatory networks, but in disease, is that you can really multiplex multiple donors in a single cell experiment, which really drives the cost down dramatically. So in a traditional bulk RNA, you would have to pay for one sample, but now with single cells, you can actually multiplex multiple donors, driving down the cost. As I mentioned, you can do perturb-seq with CRISPR. I gave you an example of pooling different transcription factors in a single experiment, so that's going to drive down the cost very much. And of course, we can not only reveal the cell type of one, but with tissue, we can reveal the heterogeneity. So the cost per cell type is much cheaper by single cells compared to a bulk strategy. So I hope that the theme that I wanted to share with you today was really scaling up, single-cell genomics for science and medicine, and hopefully this will further continue our efforts through Human Cell Atlas, and hopefully there are strong synergy and collaboration in India and Asia to further expand our efforts to map the Human Cell Atlas. So with that, I really want to thank the initial TCRE Atlas, and Shin gave a really nice talk yesterday with Jonathan, who has a poster, and Kono did a lot of the data preparation, and Piero, who's here in the audience, and Yoshi as well. Also want to thank my lab in Japan. I still do have a lab, although it's slowly decreasing, but many of the work that I've shown today were done by Joachim on the left, Kono and Yoon, and many others in the lab. But also want to thank my current lab in Singapore for their effort. And our mission in my lab in Japan now, in Singapore is, what are they made out of? What are the cells made out of? We already addressed that now with, using 5' strategy. We mapped the transcriptional network. But, we now are moving into when. I think the question is, when do they go astray for more accurate diagnostics? Then how can we control their cell fates by either small molecules or gene manipulation? So we're moving more into therapeutics, and ultimately, how can we target these cells? How can we actually deliver these drugs to these cells? So we are also very much working on these aspects in the current lab. So if you have any questions or are interested in discussing in these topics, please feel free to reach out. So with that, thank you for your attention, and happy to take any questions. Thank you.
Fantastic summary, Jay, of history of single cell and some new trends that we should all pay attention to. Already people lining up, so I don't know who was first. Gary.
Hi, Jay. Hi, hello. Hi,
yeah,
Jay.
We can hear you.
Great talk. I like the idea about mapping out the new regulatory motifs on the 5' end, and people have been doing that on the 3' end as well with alternative polyadenylation. I'm wondering if you've looked at that, and also maybe any types of relationships between 5' and 3' sort of regulation.
Yeah, so obviously one of the common tools that are popularized, like CINIC, is a common tools that people use to infer transcription factor binding sites, and that allows us to infer what could be the potential transcription factor that regulates that gene expression. There are other tools that can be used to understand the upstream transcription factors. I think the limitation there is that the 3' activity is barely based on gene expression, where using 5' strategy, we can actually narrow down and infer the promoter binding site regions based on the TSS, and creating a window and ask what are the transcription factors that may bind to that region. And that's powerful if you also have alternative promoters, right? Some regulatory of one promoter can be switched to another promoter, and this switch is mediated by different transcription factors. But if you use 3', you won't be able to distinguish the two. So it just gives us a higher accurate mapping of potential binding sites of these transcription factors. And then we can also further infer at the enhancer regulatory elements where 3' will not be able to capture. Yeah.
Lucia?
Thanks, Jay. From the virtual audience today, Mujtaba Panjapur has asked how single cell has finally been reflected in diagnosis and treatment.
That's a really, I think that's everyone's dream here. Can we use single cell as a diagnostic tool? And as a scientist and researcher, we like to believe it's yes, but on ground, in reality, when I talk to clinicians, it's very far away from reality. And the reason for that is twofold. One is cost. It's too expensive to charge patients thousands of dollars just to diagnose. So we have to reduce the cost. And only way to do that is qPCR or other simple biosensors. Again, a classical example is ART kits, antigen rapid test that we all took during the COVID pandemic, that you just do a nasal swab and you get a band or not. That's a level that we have to get to if you really want the diagnostics to be widely used. But can single-cell genomics be used to identify biomarkers? Yes, and I think that's an effort that we should strive for. Hopefully, if the cost goes down, the second aspect that why it's very difficult is there's too much data. No clinician wants to do bioinformatic analysis. That's the reality on ground. So we have to make it easy for them to interpret and come up with very easy solutions for them to diagnose quickly. Actually, more is not better for clinicians.
Two quick questioners here.
Yeah. Thanks for the fantastic talk, Jay. This is Rakesh. Actually, I was wondering about going from the single-cell data to constructing this gene regulatory network. Often that's a tedious task. For newcomers in that area, would you recommend some methods which you find useful?
Yeah, do single cells. The reason why I actually did that is I didn't want to test all possible combinations of transcription factors to see which one are useful. So that I said, why don't I just pool everything and do single-cell RNA-seq and let the empirical data tell me what are the key important genes for that regulatory network, rather than using computational predictions. Of course, computers and bioinformatics can give you many, many candidates, but these are still predictions. And until you actually do and test them in the lab, you actually don't know which one are really critical. And only way to do that is pool them, or not only way, but one of the ways to pool them and test it in the lab and see which one are actually functionally relevant. And I like to say this even for AI community is coming up with all these predictions, but you cannot say it's good until you actually go to the lab and test them in the lab. So I hope this is a learning lesson for all of us, is that computer and AI can take you only to this far. We will still have a job as long as you can hold the pipette. Yeah.
Hey Jay, I'll keep it short. But I was wondering, so the 5' capture is really good for promoters and alternative promoters. Obviously, gene transcription is really important in terms of enhancers as well, and that could be megabases away. So I was wondering, can you speculate on methods that you can combine, say maybe ATAC-seq with some of your capture techniques to kind of understand gene regulatory networks at a distance, basically?
Yeah, so we were not able to do in the same cell, but we've done single-cell ATAC-seq and 5' and overlap the open chromatin to the transcription activity. I have to say, not all open chromatin are transcribed, right? So even open chromatin and enhancer, many of them are not transcribed. Now, question is, what does that mean? Does open chromatin and then transcribe, is it useful? Is that a passive enhancer? Is it active enhancer? And I didn't have time to show this, but we have reasons to believe that transcribed open chromatin enhancers are much more associated with genetic variants that are to a disease than open chromatin that are not transcribed. And we are doing some CRISPR validations to suggest that active transcription is an indication of active enhancer versus a non-transcribed enhancers. And we hope to also correlate that with Hi-C data and see whether we see more active interactions only in the transcribed enhancers versus non-transcribed.
All right. Thank you so much, Jay.
All right.
I just one closing comment. I'll make a provocative comment. Maybe everyone should switch to 5' single-cell RNA.
Yes.
All right. Thanks a lot.
Piero says yes. Good. Thank you.
First, thank you very much. I regret not seeing everyone. I definitely have a little bit of fear of missing out, seeing the camera pan across the audience and many familiar faces. So again, thank you very, very much for the invitation and also accommodating some flexibility and allowing for me to present to you all this morning virtually. Let me pull up my slides here.
Jonah, if you'll give me 1 minute to introduce you.
Sure.
Okay, great. So as I mentioned, Jonah is Senior Program Officer and lead of the Single Cell Biology Program at CZI. So if you know one person in the single cell field, Jonah is a person you should know. And you already know him probably alongside Aviv and Sarah, the most well-recognized person in the field. And I was going through his history just now and was struck by how he's covered all the areas of expertise you really need to do the job that he does. He actually started out in litigation, in IP litigation as an intern and then as a paralegal, even before he did his undergrad, but biotech IP. Then did his undergrad in biology and then went to Duke University, did his PhD in cell biology, then went to the Salk Institute for his postdoc, again as a biologist. But simultaneously with that, so I think, as an undergrad, he also, I guess, had a side job making educational apps in Palo Alto, even though his undergrad was in Massachusetts. So Jonah was an expert at being in multiple places at the same time, even then. And then as a postdoc, he had, I think, just after a job in, just after a job in a regenerative medicine company with organoids, I guess, and bioprinting. So very much applying biotechnology for translation. And then he moved to CZI and brought all his expertise in IP, cell biology, biotechnology, translation to lead all aspects of CZI single cell program, from partnerships to technology development to platforms and research funding. Jonah, very excited to have you here.
Thank you very much. Very, very generous introduction. And maybe just to summarize, I've always been fascinated by cells and cellular heterogeneity, and in particular, the application of different technologies and engineering disciplines to those questions. And so for the last number of years, it's really been a privilege to work at CZI, and the vast majority of my team's work and our focus has been on supporting the development of the underlying technologies, perhaps the most important technology here, which is the reference atlas being developed and championed by the Human Cell Atlas community. So what I'd like to talk about for the next 10 or 15 minutes or so, and leave time for plenty of questions, are two really important frontiers in our mind. One is the diversification of reference tissue samples, so the data that is going into the Human Cell Atlas, and then starting to think more and more about how this data corpus, and I think Jay's mention of clinicians and overwhelming amounts of data, and perhaps we're all clinicians and to varying degrees all feel a little overwhelmed by the amount of data. And so how we as a community, and we start to begin to think about the advances of use of single cell data. So before I get to that, let me take a very brief moment. Hopefully I can advance my slides. There we go. To introduce you to the Chan Zuckerberg Initiative, which some of you may be more or less familiar with. So the Chan Zuckerberg Initiative is now just about 7 years old in its science goals, and our founders, Priscilla Chan and her husband, Mark Zuckerberg, founded it with the mission to cure, prevent, or manage all disease by the end of this century. And what this really is a statement in exponential growth and progress, and a focus on technologies and foundational resources, such as single-cell biology and the Human Cell Atlas. And what we are focusing on as a North Star for the next 10 years is advancing research, but in particular, advancing research to understand the mysteries of the cell and how cells interact within systems. So hopefully with this context, you'll appreciate why such a strong support for the Human Cell Atlas, working both with the scientific community, but also many other funders that are supporting this work, as well as now thinking a lot about the applications and the technologies that will flow from it. And how we're approaching this is perhaps a little bit different than some other organizations that you all may be familiar with. And in particular, a kind of multi-pronged approach that we take to supporting science in different areas that we work in. This includes funding of work around the world, and I think many grantees, both past and hopefully future, are in the audience, as well as working to support our grantees as well as the community at large by building scientific software primarily and open source tools to help accelerate growth, and increasingly now active efforts to do science via a growing network of research institutes. And specifically, this is the Chan Zuckerberg BioHub and now the BioHub networks. So let me focus on the single cell biology program, which is the one that I'm responsible for and spend the vast majority of my time thinking about. And so, as I mentioned, a large part of what we've done and have been focused on is support of the Human Cell Atlas and creating a reference of all cell types in the human body. But at our core, what we're really interested in doing is making it easy for biologists to understand the function of cells and begin to functionally interrogate and measure them. And so the focus and our vision over the last number of years has really been thinking about supporting HCA and other initiatives in compiling comprehensive data, both in health as well as more broadly in disease, that can be widely used to rapidly understand these mechanisms. And so the way that we've done this has been through a series of funding mechanisms. I don't have the time to go into all of them. I'll try to double click on just a few here to make this first point about the importance of diversification and the tissue donors. And then also to complement that funding with the building is a growing suite of technology tools that supports the accumulation and exploration and use of single cell data in a standardized and interoperable way. And this is a platform or suite of tools now that we refer to as Cell by Gene Discover that I'll come back to. And then the second and last point about driving the use has been a dedicated funding opportunity. This is perhaps foreshadowing to a final slide, which is that we have an open cycle 3 here that is open for applications from around the world, in how openly available data sets can be used to gain greater biological insight. So this could, of course, include the compilation of atlases, but also any myriad of tasks in terms of looking at gene regulatory networks and enhancers, et cetera. Again, a lot of this work, although not all, has really been focused on supporting the compilation and now hopefully the use of the Human Cell Atlas as a resource. Okay, so let me talk about ancestral diversity. This, I think, will be a slide that others have seen. This is fortunately not reflective of single cell data, although the problem persists, and that is the lacking ancestral diversity in many genetic resources. So in this example, a 2019 snapshot of the GWAS catalog. And not only is this just bad for science, but it really limits those downstream applications, again, both in the clinic, but also more broadly, in terms of driving towards precision medicine and also ensuring that this incredible resource is powered and useful and representative of the global population. So how we've been thinking about trying to make some dent in this, and certainly this is not going to solve this issue, but hopefully it's something where we can push forward and also encourage more scientists, encourage more funders, encourage others involved to work as well, is 2 active programs. The first is the Pediatric Network for the Human Cell Atlas, and the other is the Ancestry Networks for the Human Cell Atlas. In both cases, the goal was to fund international groups. So these grantees are on essentially every major continent and in about 30 to 40 countries around the world, with the explicit goal of increasing the diversity of donor tissues and underlying data across many different tissues and organs. It also includes a large component of community engagement, community input, and the goal there is both to make sure that we are being responsible to the communities that are contributing tissues and samples, but also in thinking about building trust so as to encourage the engagement with science, not just in this project, but in many others going forward. Now let me switch gears from data generation to data exploration and tell you very briefly about Cell by Gene. Again, happy to return to any of these topics via Q&A. So Cell by Gene is a hosted data platform. There's also a local tool. You can see the website here. I encourage you to jump on your computer and check it out if you would like to follow along. It is free, it is open source, it comes without limitations, and all of the data that is contained in Cell by Gene Discover that you will find is also curated into a standard enforced schema, which simply means that the data is interoperable and can be used in a more collective way such that you and other researchers don't have to go around to various different resources and then go through that often arduous and painful process of needing to reformat or wrangle data into a vast array or different schemas. And what the data looks like currently is a corpus of nearly 60 million unique cells, the vast majority of which are human. There is a growing amount of mouse data sets as well. I would highlight that it includes many different single-cell technologies. While 10x and other transcriptomic data are the most common, it certainly can support technologies like 5' or even other spatial data sets. And really an important area of growth that we're focused on in the coming months is boosting this multimodality with a particular focus in spatial data sets. It's important also to note here that, of course, grantees within CZI have contributed to this, but frankly, the vast majority of data actually comes from the community and other funded efforts. I know that Caroline Hutter and others from NIH have participated, and we've worked very closely with organizations like NIH, with the Wellcome Trust, like the Helmsley Charitable Trust, and others, and really trying to support the Human Cell Atlas and these principles, kind of a federated model that brings together the community and scientists, and this provides such a platform. Just to give you a quick snapshot of what you would see if you did go through to Cell by Gene, you would find individual data sets. They can be easily filtered and viewed based on some of those consistent metadata features that I mentioned. And so you can get down to exactly the data sets that you're interested in. And then upon finding one of those data sets, you can quickly either download that data set, either in R or in Python if you would like to reanalyze it in whatever toolchain you would prefer. You can also click on the Explore function, which would open up an interactive UMAP plot, and it can be performant and allow you to perform some basic tasks like differential expression and others in data sets now up to about 4 million cells. All of this, again, is free and open for anyone to use. One final feature that I'll highlight is a feature that we call Cell by Gene Census. So this takes all of those individual data sets currently of dissociated single cells and concatenates them and provides an API for data scientists and computational biologists that want to analyze either a single data set of now almost 40 million cells, or to slice and dice it based on different features that you would be interested in. So if you wanted all of the mouse data that are cells that are specifically labeled as microglia from female mice, you can perform that query on the order of seconds and then quickly export it into those tool chains. Or if you're looking to train models, we've developed a series of PyTorch data loaders that allow you to take this data and get it right into different machine learning toolchains. Okay, so let me end then with a funding opportunity, as hopefully many are interested and excited to participate in this. And this is a call that is currently open. I regret to say it is not open for too much longer, but hopefully long enough for those that are excited and interested. Our application formats tend to be fairly condensed. This is a call, the third cycle of data insight. So it's really focused on analysis and developing either greater biological insight from data, or thinking about different tools and data sets that can, tools and analysis methods that can help you and the rest of the community get greater insight from it. With that, I again will thank all of you for your time and the invitation. Here's my contact information as well as some more resources for CZI if you want to reach out or have additional questions that cannot be addressed in the question session. With that, I'd be happy to take some questions either here, or feel free to email me or put them in the chat. Thank you.
I don't think, yeah. Hi, Jonah. So the efforts from CZI have been really great, working towards diversity, and been immensely successful, as we can see in this conference. However, there's still a large amount of genetic diversity, which is yet to be explored, especially in Southeast Asia. The Indian subcontinent is housing about a fourth of the world's population. So is there any plans to go the Gates Foundation way, where you have country-specific calls or calls that encourage collaboration between the global south that face similar problems, to sort of build a program which is not only sustainable, right, so that the researchers get trained in the regions and they have ownership of the data, and more importantly, there is this sense of pride into working on things that are done for the country, by the people of the country, and taking it forward in the larger context.
Yeah, it's a really good question, and this is something that we've put quite a bit of thought into. I will say we have not necessarily considered country-specific calls. We have funded projects that are country-specific, but honestly have biased more towards opportunities that allow for those country-specific teams, but also enable kind of international networks of scientists and teams to come together. And so in terms of calls, we've kept things quite open in that regard. So, nice examples of this, of course, would include the IDA project that Shyam and Jay have led, and now many others. I know Partha, who's hosting, and others are involved in. So that's kind of one answer to a question that I heard. In terms of then the opportunities that I think Gates is exceptionally good at in building knowledge and knowledge exchange and training opportunities, this is something that we have done some of, and there's been some early pilots via the HCA Equity Working Group in terms of trainings, both for data generation and also analysis. There was an initial grant and project there that I think has resulted in 3 or 4, and John and Lucia may correct me there, incredibly successful projects. And I think, on behalf of CZI, as well as I know a number of other funders that have seen the success of those, I think doing more of those is something that we will absolutely be interested in pursuing. I think the answer is, we've done bits of this in kind of different vehicles, not necessarily thought about doing it all in one shot.
And one suggestion, as the cell cross gene gets accessed more and more, I can talk about an incident that happens in my classroom, is when 50 students are trying to access it at a go, there are certain latency issues. So just like UCSC browser or some of these tools which are used, it would be great if you have like a mirror that is hosted in this part of the world so that the latency is reduced so that we can actually use it in the classroom and get more people working on it.
Yeah, please reach out. We've heard the use of the tool in the classroom is not necessarily something that we initially designed it for. We've been hearing more and more about this, and honestly are extraordinarily excited and interested in what that looks like. And latency issues are like catnip to engineers. So please reach out. You'll make all of us very, very happy and appreciative of that feedback.
Thank you.
Thank you.
A question following up on what Ishan said. There's no doubting CZI's commitment to increasing diversity in the Human Cell Atlas. Like you said, the pediatric networks is age diversity, and ancestry networks is ancestral diversity. And that has really been transformative in planting seeds all over the world to diversify the HCA more than I think any other funding initiative I can think of. Now, you have mentioned often that CZI cannot, on its own, do everything in the funding landscape. By definition, there isn't infinite funds, and that's why you're on the Funders Collective for the Human Cell Atlas and coordinating with NIH and other funding agencies. Yesterday, Carolyn Hutter was from NEGRI, gave another talk similar to yours, but she also made a strong commitment to expanding diversity and how scientifically that was important for the research. I put it to her that that recognition came at least in the mid to early 2000s for the human genetics field, but the needle has not moved since then. In the 15 or 20 years since then, there's been zero change in the amount of diversity in GWAS studies, which is sobering. So I'm sure even then the funders and the community were intellectually and even morally just as committed, but for one reason or another, very little happened. And I'm wondering if there's a discussion of this point in the Funders Collective. Is there a recognition that, or some discussion of what went, to put it bluntly, what went wrong over those 15 years for human genetics diversification that we can avoid in the single cell field? And 10 years later when we come back, things will actually look different.
So no, there's not necessarily been a discussion of what went wrong. but certainly an acknowledgement that something went wrong, right? And that this is a problem that needs to be addressed. I am simultaneously very proud and very excited of the work that the community is doing via the pediatric and ancestry networks, while also admittedly, nervous, knowing that we need to go much, much further. And that is something that I think as funders, as scientists, we really need to advocate for, both in terms, again, of the importance of it for society, but also the importance of it for science. I think it is projects like those that are at the front and at the vanguard, like IDA and like others, that will establish that precedent, already have established that precedent, and one that's going to be incredibly important to really champion and elevate and share to build more excitement and build more interest. So I don't know that fully answered the question, other than I think that clear acknowledgement and that clear commitment, again, from CCI, but again, other funders, it's wonderful to hear that Carolyn expressed a similar statement, because certainly the resources and the reach that organizations like NIH have are really where things can go to a very large level.
Thank you, Jonah. Thanks. Partha.
Jonah, you have done, as CZI, you have done exceedingly well in bringing groups from different countries together to work and on the single cell projects. But again, like you pointed out, that even though you have an eye towards internationalization, bringing people together, funds are not infinite, and therefore we also need in these various countries to put in some money towards these kinds of research. My question, and this question I also asked Carolyn yesterday, my question is, do you engage with funders in other countries and sort of promote these kinds of activities such that national funding also increases? Do they have an ear? Do you do the advocacy? I think that's very important, because we all can't completely depend on CZI and bodies like the CZI NIH to do our own work, and it'll help us in it'll help countries like India or scientists in India in a big way if you can provide the advocacy to Indian funding agencies.
Yeah, it's a great question, Partha. I think different views of advocacy are differently, but certainly have spent a good amount of time looking for opportunities to partner and complement efforts. I don't know, at least for myself, I don't think that we have ever engaged or spoken with funders in India, but certainly have in other Asian countries. Japan comes to mind, as well as a few others, and then certainly in Latin America. I think in most of the cases, my experience has been that actually the complementarity of efforts is very high. And that gives me hope in the future that these discussions and these opportunities and ways to work together are likely abundant. And in particular, kind of going back to our mission of thinking about technology and fundamental resources. And in many countries, especially national funders, the remit and what they owe their taxpayers in terms of disease research and really thinking about applications are nicely coupled. And so this is simply a statement to say that I think in the past we have had these discussions. I think it takes a lot of time and a lot of trust to be built, and certainly something that we'd like to do more of and are looking to, again, in these next 5-10 years, broaden that international reach, find more opportunities to work together and find big problems that we can complement and partner with domestic, other foundations, and other funders regionally.
Thank you very much. Do that for our sake.
Introductions are helpful. Yeah.
Thanks again, Jonah. Wonderful to have you in the meeting.
Thank you very much.
Over to Wong Yang for the next session.